We are live on Twitch, so if you want to start the introductions.
Hello, hello everyone. We'll just give everyone a few minutes to trickle in, but we'll get started briefly with introductions. Can you share your screen, Jane? Maybe we could start with the cover slide. So for those who are joining for the first time, this is a journal club we try to host every 2 weeks, and sometimes we're lucky to have an author join, as we are today. And really it's an opportunity to sort of have an interesting discussion on a topic with a paper in mind to help us frame the conversation. But if the conversation takes us to an interesting tangent, we also welcome that. But we are going to try to make it through all the figures and save some interesting points for the end as well. And today we have 2 special guests. Brad English is our our special co-host today, who will be helping us lead the discussion. And we actually have the PI on the paper. I'm sorry if I'm mispronouncing your name, Olivier, and I'm sorry I can't say your last name properly, but thank you so much for joining us as well. And we're going to frame the conversation about getting to know Brad in the first few minutes and sort of how his interests have changed, or how his interests have molded from aging to also developmental biology, and that'll kind of help us frame the context of the later discussion. And then we'll kind of dig into the meat of the paper. And again, we're all PhD or MD PhD students across the 2 coasts, West Coast and East Coast. Feel free to join our Slack. DNA Deviants is linked on the Twitter. If you want to continue asynchronous brainstorming conversation, or if you just want to join hopefully a fun biotech community. We host some events in person, but most of our events are every 2 weeks online on Twitch, where we go through a synbio or longevity paper. And so moving on to next slide. So again, welcome Brad. Brad is currently a PhD student at Harvard.
Yeah, so this paper deals with looking at how and why different organisms develop at different speeds from sort of conception to like birth, I guess. And what's really interesting is that different organisms have different rates, even though the sequence of events that happens in development are very similar and highly conserved across organisms. So if you look at how a mouse develops versus how a human develops, they'll go through the same path more or less, and it'll also kind of look very similar across most of those time points. But the speed at which that happens can be very different. A mouse sometimes takes around 20 days, whereas a human takes around 9 months. So why does this happen? That's basically the question that this paper sought to answer. And one possible cause is the difference in sort of underlying biochemical reaction rates. So let's say like all of the different things that need to happen just happen more slowly on their own timelines, and aggregate of those things causes the overall development time to be slower. But this paper basically shows another reason why this is happening, and this is really interesting for regenerative medicine. If we can go to the next slide. Yeah, so basically regenerative medicine is a way to try to get around a lot of the problems of aging, not by slowing down the aging of an individual patient, but instead taking a patient who's having some kind of problem with an organ and extracting a stem cell and using that stem cell to grow an organ on the spot for that patient to just like do the transplant. The big problem here is that growing that organ will take a lot of time, and often these patients don't have time to wait for 9 months for a new kidney to be grown. And so a kind of underlying technology that would need to be developed for this is a way to modify and speed up the developmental rate of the growth of these organs. And so that's why this kind of research is really interesting and important for possible sort of interesting therapeutic potential down the line. Yeah, I think this was.
Yeah, I can jump in here. Sorry. Yeah, I'm having a little bit of internet issues, so I apologize if someone expecting me to jump in earlier there. But this is just some kind of dotting the space around this paper a little bit here with some random facts that we can think about, and we can dive into a little bit more deeply as we go further. But it is the case on the whole that other than species interspecies differences like smaller dogs versus larger dogs, that larger animals tend to live longer, and bigger animals tend to have lower metabolic rates. They also tend to be somewhat more efficient with the energy that they use. For instance, like a giant squid can eat like a very small fish relative to its body size, but still use that energy efficiently to keep it going. And you can go to the next slide. And so I think Olivia would be better to sort of explain this slide far more than me, but I think this video is very important to watch because it shows this body elongation phenomenon that happens very early on in embryogenesis. It gives rise to a whole wealth of different tissues. But as you watch it happening, you'll see that there's this cyclatory, if you read the paper, you'll be somewhat familiar with it already, these oscillations that occur of different, in this instance, you're seeing a fluorescent dye show up at a particular point that has a very regularity to it that happens over the course of the cycle. And it's different between mice and humans specifically. In mice, it happens twice as quickly than humans, and this is kind of sets the sort of stage for a lot of the investigation here in this paper. Why does it happen faster to that extent? This sort of segmentation clock, it's called in the paper, is a very nice hallmark or light showing on development itself. If you were to look at how fast development would go in some context, this is a very well-supported place to look as to how the rate and speed of it is occurring in one species versus the other. It's a beautiful video too. There's a lot of other beautiful videos that come from the lab as well as are in this paper. You can go next slide. This was not mine. Does someone want to pick up from here?
So a nice transition point is to kind of jump in and sort of lay out the groundwork for how this paper kind of takes on the feat of trying to sort of understand what could be differences between species, cross-species development. And so it's using in vitro models, and I would say like the crux of sort of a fundamental point that we need to make and spend a little bit of time understanding is sort of what this segmentation clock is. And I think I'll hand it off to Brad to kind of set the stage, but I think a better way to do it will be to sort of jump in to the figures directly. But in sort of digestible terms for those listening, how would you sort of explain what the segmentation clock is?
Well, I mean, I feel like I could make an attempt, but in the presence of Olivia, I feel less. I'd definitely like to hand up the mic to him to share that with the public.
I'm not sure. Tell me if you want me to jump in. I'm happy to do so. But I thought this is your.
Yeah, it's the rate at which these somites. If you're familiar with somites, and these segments that sort of extend beyond the tail-like structure at the end of this long structure that happens following gastrulation, and sort of like budding out segments that appear. And the most sort of like intuitive things associated with those are vertebrae in the spinal column, but these give rise to a lot of different things. But they're these like chunks that kind of sort of elaborate themselves into the segments as the body extends for the very first time over the head-to-tail axis that it's going to become. That's a brief introduction, but again, Olivia could give a much better explanation than I can.
Oh, sorry.
Do you want to add anything to that, or
no? No, I think that's. I mean, the clock is basically an oscillator that controls the rhythmic production of the somites, which contain the precursors of the vertebrae and the skeletal muscle. I think what's important is that it oscillates with a defined period, and this period is specific for different species.
But that's what we're using here.
What was your thought process when you were designing the clock? Like you were setting up to do an experiment, you're thinking about all these different questions, and what made you decide on making this particular clock in this way?
So no, we did not design the clock. So we identified it quite a while ago, actually, just by looking at chicken embryos, and we saw that some genes were expressed with a complex sequence, which appeared as a sort of transcription wave that was sweeping along the precursors of these segments, the somites. And this wave was reproduced each time a new somite would form. So it was known that somite formation was a periodic process, so that the somites are, as we said, the precursors of the vertebrae, and so the embryo will form a defined number of somites, which in human is 55, but this proceeds with a very defined rhythm, which is species specific. And so what we found, this clock is a molecular oscillator that's basically a gene regulatory network that triggers periodic activation of genes in the precursors of the segments, and in fact, which controls the periodicity of the whole process. So if you want to think about, because the whole problem is to form a segmental series, like a periodic series of anatomical structures like the vertebrae, and the simple way to think about it is as if you were displacing an oscillator in space, you would get such a series, and that's actually what happens in the embryo. Is you have this oscillator, the clock, that gets displaced in space as the embryo elongates, and that's how you basically make these periodic anatomical structures.
I guess that kind of makes sense from a development or evolutionary point of view, that it would be easier to evolve just like the ability to make one vertebra and then like to expand that out by just repeating that process over and over again, instead of needing to like evolve something new for each vertebra that you're trying to evolve into, if that makes sense.
Yeah, and it's not only vertebrae actually, because it's now there are evidence that there is such a clock also operating in invertebrates like worms, and which also that's exactly the same idea. They form these metameres, which are complex anatomical structure, but they reproduce these metameres for a large number of units.
Thank you. See, I told you to do it better.
So Jane or Kat, did you want to talk a bit about Kleiber's law here?
Oh yeah, Jane, did you have a point to add for this slide?
This was totally new to me, so I just quickly added this in for just more context. But this is fascinating to me, as I'm an immunologist, so I don't really study developmental biology or metabolism. But this was interesting for me to find out that for most animals, the metabolic rate is not proportional to the mass, but it's the theory force power of mass, which was really interesting. And then Brad gave some more interesting examples of different lifespans in different species. But I thought this was interesting too. And actually, when I read the paper, I was surprised that there were not more extraordinary differences between a mouse and a human, just because I think like mice live like 2 to 3 years, humans live 100 years. I thought there actually be more substantial differences between the two, but it was kind of interesting that it wasn't as great of a difference, at least from my naive perspective, as like not in this field of study.
Another striking example that people use often is to say that the heart beats the same number of beats in mouse as in human, knowing that the mouse has a lifetime of 2 years and human is about more 80 years. Means it beats much faster.
I think this is where kind of like anomalies or animals that don't kind of fit nicely along this model are interesting cases to pinpoint. Like bats are to me are very interesting, and heartbeat is one. Like they have a much faster heartbeat based on what would be expected for their body mass. And there's very clear like life's interesting cases. Folks who study longevity, methylene comes to mind of just like comparative genomics and bat species specific differences. So to me, like this was like the moment where I was like, okay, this is now I want to find anomalies and kind of do a similar comparison there of how they maybe have very different developmental paradigms.
Yeah, one of my favorites that still boggles my mind a bit, that in mammals, if you have the diameter of the aorta, you can immediately predict the size, like on average weight of the mammal itself. It scales as a space-filling model. Apparently, they've done the physics on that. Found it's always within a certain very small error margin. Found that fascinating. There's some conserved laws here.
So let's move on to figure 1.
Yeah, let's do it. All right. So the first figure is sort of kind of setting the stage and on a couple different ways as regards to what is different between these species, and more notably, perhaps what's not different, or what can't be the possibility that would explain why there is this difference in developmental rates. So developmental rate itself, as here indexed as the oscillatory period of the segmentation clock, is twice as fast in mice, as you see in figure 1A, as humans. Again, as Olivia mentioned, this is the rate at which these sort of segments, somites appear one after the other. Notably also, the amount of this marker for an actually differentiating, shown in figure 1B, is higher in mice early on. And then higher in humans later on. But one of the things in figure 1C you'll see is cell cycle duration is actually slower in mice. It's faster, so it's shorter. And this is one of the things that's already very early on kind of set aside as not the possible explanation for why there is this difference between the developmental rates. This is one of the craziest things that I'm obsessed kind of with some transplantation things that happen a lot in developmental biology, and this is a really nice one. But they found that when you culture the human cells with the mice, or vice versa, at a ratio of 1 to 100, so they're just surrounded by the xeno other, they actually don't change the segmentation clock. It's embedded into them in a cell autonomous manner, and that's what you're seeing in F. Let's kind of set stage. Is there anything more that you would add here, Olivia?
But one thing I can add is that one thing we knew is that if you take human cells, like differentiated to this stage where you have oscillations, and you culture it in isolation, they will, if you treat them with, in fact, a YAP inhibitor, so the cells will oscillate, and they will oscillate with their normal period. And you can do the same experiment also with mouse cells. And so, which means that, and that's in complete isolation. And we don't understand how this can work because these oscillations are supposed to be driven by notch. But the experiments I'm telling you about, there's no cell contact, and yet they keep the endogenous period. And so that's something that is still quite mysterious. But that's why we are not so surprised to see that in chimeric conditions that would keep this this period, because it seems to be really hardwired in the cells.
I have a technical question here. So in terms of the culturing conditions for the two different types of cells, were they optimized for what would achieve the best, I would say, proliferation growth happening to the cells for the individual, the two cell types, or was it like how are those conditions chosen? Because that actually makes a huge effect of just like media and quantity of what you add to media and proliferation rate, even just like in traditional cell lines. So I'm curious, like if for something that's like super time specific, how you sort of like make sure you had the right paradigm or the right composition for that.
Oh, that's a very good question. The whole difficulty actually of this was to find identical culture conditions that would work for both species, because if you start to play with the goodies and particularly the nutrients and so on, then you're going to bias the metabolism of the cells, and so it was critical to find conditions that would work for the two species. And because I think this experiment, which is described in the paper, is possible because we use a pure population of cells for both species, so they both sorted for this marker mesogenin 1 at exactly the same time, developmental time, and in exactly the same culture conditions. So there we can really compare, the differences.
I guess like the question though for me still that I don't fully get is how did you kind of benchmark that this was like an appropriate condition, that both cell types were happy? Like what were sort of the criteria for like this is an appropriate culturing condition, or like one that isn't somehow perturbing the cells?
But there are two elements. One is that you get the maximal induction of the fate, this pre-Semitic mesoderm fate. So which so in mouse it's a bit less efficient than in humans. But so Margaret found conditions where she would get 80% mouse cells and 95 or even more human cells, but she still sorted the cells by facts. So that's one criterion, and then that of course that they recapitulate the oscillations efficiently.
That's very helpful. That's very helpful. And yeah, that's so beautiful. The co-culturing experience, especially as Brad alluded to, is a very kind of very striking, like that result, especially because like you have maintenance. That is like an intrinsic signal that you have maintenance of the clock, even in absence of in a co-culturing system.
Is just background, I guess. Is this type of co-culture experiment a common experiment in developmental biology research? It sounds really clever a way to kind of naturally probe whether something is being driven by internal cellular events or like crosstalk between cells during development.
Yeah, it's a very classical paradigm of developmental biology where you put, say, you can do this with embryos. You take a tissue, for instance, you put it elsewhere, and you look at the fate of this tissue, whether it adopts the fate of its new position or whether it keeps its intrinsic fate. So conceptually, it's very similar experiment, except that it's more in vitro.
Really cool. Thanks.
And if you want to see something very horror show, you can look up axolotl limb regeneration and look at all the places they've replaced those.
Yeah.
Oh yeah, so now we're looking at all the different possibilities of why this period is different between the two, and normalization is obviously key here because mice cells and human cells have, as you see in A, different volume, and B, different mass. When you correct for those two, you get C, twice the oxygen consumption rate, or OCR, of mice versus humans, and there's also twice this extracellular acidification rate in D. I think I'm getting that correct, where this is indicatory of glycolytic rates, and carbon dioxide secretion. So I think this is what sort of started pointing in the direction of something going on here with regards to metabolism, as once it's corrected for between the size and mass difference, both wet and dry, they did in these experiments, but there is a difference in that in particular.
So here I should, just for the story of how things happened. Initially, because the measure of the oxygen consumption rate was done with using a seahorse instrument, where you use spatial plates that are seeded with a defined number of cells, which is what Margaret did to compare. So she compared the same number of cells for human and mouse cells, and she found the same oxygen consumption rate, which was very surprising because we were expecting a faster one. But she was following the instructions of the seahorse. There was no reason to think that the cells would be larger. I mean, and when you say human cells are larger, that's not true for all cells. That's true for these ones. But for instance, in the paper, she also looked at neural cells, and they're not larger. They're the same size, but their metabolism is more active. And then Margaret, just by eye, she realized that the cells were bigger in the human whales. So she started to really measure precisely the volume and the mass and all that. And then she realized that the human cells, this type of human cells, were twice as big as the mouse. So you had to normalize by mass in order to compare the data. But it's it's much to her credit to have noticed this, because a lot of people would have gone just said, okay, it's the same. And this notion of the cell size is also a very interesting one, and we don't. It's not. It should be done in a more systematic fashion. It's not. There's very little data in the literature about the cell size in different species. But it's not as simple as human cells are twice as big for sure.
That's very interesting. Oh no, sorry to interrupt. I was just going to ask about mitochondrial copy number, even since like for oxidation utilization, like if there's a way to also just normalize by mitocopy.
Yeah. So she measured the mitochondrial the amount of mitochondria using a green dye which labels mitochondria, and so there she saw that there are more mitochondria. But there again, it's also been a complicated story because some dyes are also they sensitive to the potential, to the membrane potential of the mitochondria, and so that initially she used there is a mitotracker green and the mitotracker red, and the red one I think it's sensitive to the membrane voltage, and that was completely confusing the results, and she didn't find initially that there was this increased amount of mitochondria, which makes complete sense, and but the way you just label the cells with the mitochondria and you analyze by facts and then you get just a measure of the total amount of mitochondria in each species.
Oh, so just like a fluorescent intensity quantification.
Exactly. Yeah.
If I'm not mistaken, you found that both more mitochondria in the mice cells and they're more active per mitochondria.
Yeah, that's correct. Because initially, we were happy with just having more mitochondria. So there's about twice as more as you can see here, but they're also more active. So that really pointed in the direction of the mitochondria as maybe a mechanism to regulate this developmental time.
Do you think there's any major biases with like doing FACS isolation of like desired cells? Like I think going back to then like the phrase, if you FACS isolate, whichever population you want, you're kind of selecting for the most like robust cells. Since it is like it is like a pretty intensive process, be sorted and then be in like a very unideal condition, which often even if you're really fast, like on the order of you can't probably make that process shorter than like an hour start to finish. So you are kind of still selecting
Yeah, so that's true. I guess not all the experiments, it was not possible to fact sort for all the experiments. Some were a bit more sensitive. And in this case, we were happy, I guess, with the 80% and 95% because, that's, but yeah, that's a lot to ask, I mean, to the cells.
Yeah. No, it's not like a crystal, it's more like a consideration for the in vitro field. Like it is definitely a huge stressor that we just have to deal with because it is a good way to select. But
on the other hand, if you don't fact sort and you have contaminants, like even if you have 20% of the cell population that has a different metabolism, which is what happens, then you can completely confound your data.
Yeah, absolutely.
Okay, cool. Now here's really the meat and potatoes of where those keen observations, as Olivia mentioned, Margaret did, led them to. Notably, you can see a schematic here of a classic electron transport chain, where you have sort of electrons handed from one cytochrome complex to the next, and at each stage you're having protons pumped out to make this proton gradient, and all the while you're sort of generating a lot of these other molecules, and we'll come back to those. But first off, for each of these different complexes, when they were inhibited, it was found that the segmentation clock period actually increased. You can see that both for complex 1 with rotenone, 2 does not get attention, and then basically all of them up until ATP synthase, which didn't have a change or a dramatic one between that and the control. I believe it did reduce the oscillation amplitude though, and made for a premature number of oscillations. But sort of, correct me if I'm off here slightly, but pointing toward this sort of energy differences and the mitochondria differences led one to the mitochondria and specifically to electron transport chain, and notably that the action must be going on somewhere there. But then at a certain point with this FCCP enzyme that was incorporated here, which allows for a leaky diffusion of protons back into the mitochondrial matrix, it seemed like ATP production itself was not responsible for producing the shorter oscillation clock period, but rather there was something else going on. And that's what then eventually led to looking at NAD specifically, which you found from the sort of cancer biology field. Decreasing cellular proliferation, PDK inhibition specifically activating pyruvate dehydrogenase, and that led to which leads to a lower NAD plus NADH ratio. And that, instead of sort of inhibiting these specific complexes, was something that pointed you sort of closer towards what might be the modulating or stronger modulating factor on the segmentation clock.
Yeah, it was a bit of a surprise because initially, intuitively, you might think as regulating the amount of ADP as a good way to tune the pace of metabolism. Because one of the things which is very striking in this control of the developmental timing is the fact that it really acts on many different reactions, cell reactions, like transcription, translation, all kinds of important cellular reactions. So ATP in this sense was a good potential culprit. And then NAD, the NAD NADH, which reflects the redox state of the cell, was a bit less intuitive how this might impact the developmental timing.
Yeah, and I've included in the top right figure DCA's mechanism of action, which you included to show that it's sort of shuttling away NAD plus from the mitochondria, and it also produces a proton difference that doesn't allow the first cytochrome complex to compensate for that. And that was something that you borrowed in from or noted from the cancer biology field as something that might be an effector here. And apologize, I have something in my notes too that I wanted to mention about that, but feel free to comment on what sort of points you in that direction while I pull it up.
Yeah, in the cancer, that's a paper from Matt van der Eyden's group where they also showed that the NAD and NADH ratio was playing a key role for the control of cell proliferation, and you could act on cell proliferation by modulating this ratio. And ways to modulate this NAD and NADH ratio is what's shown on A. Like for instance, if you inhibit PDK with the DCA, you're going to activate PDH and then increase the NAD and NADH ratio. There are many different ways you can also add pyruvate to the medium, and that's also going to push the reactions towards production of NAD and so on. So there are different ways that you can modulate. What is a bit unclear is the transport of NAD from the mitochondria to, or say the exchange between the mitochondria and the cytoplasm. Because suppose normally it uses a system of shuttles, like the malate aspartate shuttle, and but recently there's been also some receptors identified, so this whole area is a bit murky. And we talk, for instance, about the NAD NADH ratio, the cytoplasmic one, which seems to be important. And here in the paper, it's measured by looking at the pyruvate over lactate ratio in the extracellular medium, which is a proxy for the NAD NADH ratio, and that's something that's been worked out by people working in the metabolism field, like Vamsimutan and others. So that's in the end what was used.
And so instead of looking at oxidative phosphorylation at large and sort of generation of ATP as the sort of key motivator or key influence on the change in the segmentation clock period, namely by including this FCCP enzyme that allows protons to go back over, you went to NAD plus and the NADH ratio. But isn't it a very, again, I speak incredibly naively here, that sounds like a very dramatic change that suddenly ruin or manipulate or mess with a lot of the ATP production that's happening in the electron transport chain. Are you restoring the ATP supply while doing that at the same time so that it's not sort of ruining a lot of the energy production that the cells need to have done to begin with?
What do you mean? Sorry.
So the
FCCP?
Sorry. Yeah, the FCCP sort of ruins all the work that the electron transport chain is doing here with regards to the proton pump. So ATP reduction is dramatically reduced. Is it being rescued some other way topically? So that's not a sort of, how's that not killing the cell, basically?
It is killing the cells. I mean, all these, oligomycin is also killing the cells. So these experiments are necessarily short term. And one of the difficulty of the exercises was for Margaret to find really sublethal doses that would elicit a reproducible effect without completely killing the cells. But if you use like a regular dose of any of these drugs, they're super toxic drugs.
Got it. Yeah. Okay.
And that's why the experiment, the gain of function, is critical. You see, because these are all these pharmacological experiments. They're very good to dissect the pathway and what's important. But it's much easier to slow down than to accelerate. And that's why it was really important to show that you can also, by playing on this mechanism in the opposite direction, you can accelerate the system.
Gotcha. Did you want to add anything or add a question by any chance?
Yeah, well, actually, it's kind of answered already. But I was just wondering, because when you were treating the cell with DCA, I think you didn't see the change in oscillation, if I remember correctly. I thought it was a little bit strange because when you're treating the cell with DCA, you are biasing the direction. You're biasing oxidative you're biasing oxidative fermentation, and this pushes the cells into forming acetate instead of lactate, right? That's I think so. And then in that sense, shouldn't you be regenerating NADH?
No, you're absolutely correct. So what Margaret showed here is that it can rescue the impact of treating with azide, which would slow down the oscillation. So if you treat with DCA, it can rescue and bring it back to the normal period, but it cannot accelerate it. And that's, of course, I mean, I don't have a good answer why. And so, but if you have a worse baseline, it gets it better. But if you treat only with DCA, it was not enough to shift the system. And that's why the only experiment is the one which could do it, is the one which is shown in FI. And but I agree. I mean, that would have been our hope that DCA would do it, and why it does not. Maybe it's not powerful enough for. But it does it. It goes in this direction by rescuing the effect of sodium azide, nevertheless.
Okay. And another question I had was whether, I think it might have been done in the paper, and I have to admit I only skimmed the paper, but have you done this NOx gene transfection in a cell line that's lacking respiration, and would that transduction itself being able to rescue the cell's oscillation?
No, that's not been done. It was only done in wild type cells so far.
Okay, because if we reason this through logic, I would also expect this enzyme itself to rescue the cell, right? Okay.
Cool. Yeah. And moving on to the last figure of the paper. Now that NAD+ was found as perhaps one of the only things, please don't correct me if I'm wrong here, that can actually increase the speed or lower the period of the segmentation clock. Many things can slow it down, as you saw, temperature being another one that can speed it up. But nothing has been found in the past, or ectopically and chemically, to reduce the period of it. The researchers looked to find out if there were other things mediating this, or what might be happening downstream of this, and incorporating these pyromycin tags onto peptides that were being actively translated between the mice and human pre-Semitic mesoderm. They found that translation was also faster between mice and humans, notably. And overall, though, this rate of protein synthesis was, the degradation was also faster in mice. And this kind of ties back to a earlier paper in the field that found out that the protein stability of animals that develop quicker is far lower than one that develops slower. So in mice, the protein stability is a lot lower, so you have a lot more turnover, but the proteasomal degradation is not as strong as a influence as the rate of protein synthesis with regards to developmental speed and explanatory of the differences between mice and humans. And so that's basically what this figure is looking at, but also it's not as it's mediated as opposed to driving the whole developmental rate, which we see more so being driven by NAD+ and manipulations therein.
I'm curious if you thought of kind of looking at specifically, which proteins, or using like a method to get like a proxy for how translation changes, like maybe ribo or polysomal profiling, to sort of just get like a translation snapshot at relevant stages, like within development, between species. I thought that would be kind of like an interesting next step along these lines.
Yeah, that's so we've been thinking about it, and these are not super easy experiments, so we started to discuss with
as I transition to, I do doing polysomal profiling not on human cells, so just getting the protocol to work appropriately in my cells definitely is not an easy feat. It's a very long protocol too, but the readout at the end of the day, like once you have the conditions you're looking at and sort of which interesting fractions you want to look at, like the readout at the end of the day is an RNA-seq analysis, so it's the pain to figure out your conditions.
No, absolutely, and that's Margaret now, so she has her own lab at MGH, and she's looking into these things. She started collaborated with Sterling Churchman in my department, who's doing this type of experiments. They're also doing this time-lapse, mRNA time-lapse experiment where they measure using sequencing. They also measure all the stages of RNA production, like splicing, and then the time. Because what she found, which was a bit surprising, is that transcription doesn't seem to scale, unlike translation. So it's not in the paper because these are difficult experiments, but the preliminary data suggests that for transcription there's no scaling.
Interesting. Is there an uptick in like potentially just RNA editing, or that's very interesting to see like if changes are happening at the translational level instead of transcriptional. But yeah, I'm curious like if there's like to what resolution you're looking at the transcription, like maybe there's like an uptick in.
Well, there is. I'm talking about the rate of transcription. I'm not talking about other, like splicing, exports. There are many different steps. So she's currently measuring all this and comparing these different steps between the two species.
That's very cool. I'm sorry, Brad, to cut off if you.
No, that's perfectly fine. That ties in very nicely to a question that I was thinking of asking the other day, which is very similar. But are there any other sort of levers and dials that you think could be pushed and pulled to further speed up development? And if so, do you think it might have sort of detrimental effects on human cells or human development if it were done either in an organoid or another context? And if not, is that because simply evolution hasn't thought of it yet, or any sort of comments on that, Flora? Give it to you.
I think that's a very good point. I think we need to know more about the mechanism. One of the things you may want to do, for instance, is if you could increase the number of mitochondria in the cells, And there might be ways to do so. I mean, there are genetic ways to play with the transcription factors regulating the total number of the mitochondrial biogenesis. That could be one way. You could also imagine to act on the electron transport chain, so the machinery itself to increase, to boost. So, all this would point in this direction, but it's tricky, and playing with metabolism is really tricky because there are all kinds of feedback. So if you do one thing, then it often elicits some unexpected consequences because of some feedback that we immediately correct, and sometimes overcorrect the modification that you've tried to elicit. So I'm hopeful that there will be ways to do it. It's still early day. I don't think we have enough understanding of what's going on to do this in a really intelligent way. The only thing that works is the temperature so far, which is interesting and speaks about probably the mechanism also.
Yeah.
Sorry, can you expand on that, the temperature?
So if you take embryos, zebrafish embryos or fly embryos, if you change the temperature, you can change the rate of development. So there is a significant, at least for all the cold-blooded animals, there's a significant window where you can play with the temperature, and that will increase the pace of development in a linear fashion, such that you could even extract the Q10 and all this, which would somehow suggest that there's a rate-limiting step that controls the speed. Even in warm-blooded animals, like in the chicken embryo, for instance, it's in my lab we work also on chicken embryos. You can just incubate them. You take them out of the incubator. They will be at a defined developmental stage, and they will freeze. They will stop developing, and then you can put them back, and they will resume development. You can slow down if you don't put them at 37 or 38. You put them at 35, they will develop slower. So, but usually this is very, it's still normal development. You're just tuning the pace.
Oh, no, that's very fascinating. Yeah, chicks are wonderful models for development. For context, I worked in Connie Sepko's lab for a few years. I just remember I took a course with Angela Spur, and you were one of the lecturers. I was for the whole time I was wondering where have we met previously, but now I just remember. But yeah, that's very interesting. I'm also curious of just if temperature is so crucial, how well we do even understand how well different parts of the bodies maintain kind of core temperature, because it's not even, I think we have global physiological temperature information of what's consistent, but not to the level of organ specific or what sort of environmental fluctuations, how that has very compartmentalized effects in the body. To my understanding, it's still a really fuzzy area, but that's something I'm really interested in my own research.
Yeah, no, it's very interesting. I mean, there are some thermogenic cells like the brown fat, for instance, which is basically doing what the FCCP is doing. So it's uncoupling the electron transport chain from the ATP production. So basically, it burns energy just and generates heat. Because it loses the coupling with the ATP synthase. So and then there are other mechanisms like with the muscles and promoting muscle activity and so on. But what controls the temperature inside the cells is not really understood. There's very few people working on this. We started to discuss with people, physicists, and try to see if we could learn more. But it's a complicated field because the tools are not really ready to the physical tools, the dyes. So there are some temperature sensitive dyes, but it's controversial and so.
No, definitely. Yeah, that's exactly kind of what I'm working on in my lab. Since one of my labs is a hibernation, we're a hibernation lab, so it's called.
Okay, that's interesting.
Yeah, but it's very fed into this conversation. It's in my own interest, but.
So where are you?
So at Whitehead, Sinisha Hervatin's lab, and also commented by Jonathan Weissman, who's doing a lot of screening, and but the overall focus for our lab is understand the drivers of hibernation and cold tolerance.
Cool. That's interesting.
Very cool. I think we probably want to open it up for questions at some point, Jane.
Yes,
but I can.
Well, in the interest of time, to make sure that we're respectful of everyone's time, thank you so much, everyone, for joining. Especially Via, thank you so much for your expertise and spending the Sunday with us chatting about this really cool research. We did have a guest hop on, but if at any point if anyone needs to leave, please feel free to hop off, and you can review the recording to those listening on the Twitch stream. And Darvesh, welcome. Thank you for joining us again. I think you've joined previously, so thank you for being a continued journal clubber. Feel free to ask your question now.
Yeah, for sure. One was kind of a pretty specific question, but in figure 1 for I think 1C and maybe 1E, I was just wondering what does the horizontal position within mouse and human actually mean? I would understand if it's a box plot, but it doesn't seem symmetric either. So it's not a perfect kind of mirror. So does that
You mean the I think it's just the middle one is I guess it's the mean, but that could be the median as well. I'm not 100% sure.
Gotcha. I guess I meant I can kind of understand how the sort of black line itself, or I guess the yellow line, the mouse, is probably the 75th percentile, 25th percentile, the median. But within human, it seems like the dots are kind of distributed horizontally as well. And I was wondering, does that have any specific meaning, or is it just to visualize?
It's just to visualize. Yeah, no, there's no specific meaning.
Gotcha. And then for figure 4, I was wondering, is it just difficult to normalize to the number of ribosomes, and also is the number of ribosomes correlated with the size of the cell? Because I guess both seem to make sense, but I was wondering if normalizing to the mass of the cell is more upstream than normalizing to the number of ribosomes.
I think it's more difficult to normalize to the number of ribosomes. Okay. I'm not sure how you would count the ribosomes. You could probably use the ribosome RNA.
Could you not, if you do a sucrose gradient and a digestion of your digestion to monosomes, and then you could look at your monosome peak just to see the quantity over, but that would be a painful process to just do that.
I think the idea here was to have some sort of crude measure of the rate, which is what you get with the puromycin assay. She also used another, because the problem here is that it's when you incorporate the puromycin, that blocks the translation, so you only generate peptides. But she also used another kind of labeling, which we're using AHA, which I think is an analog of methionine that you can also label with an antibody, and there you don't make peptides, you really generate the entire proteins. But it's not protein specific, so it's really a measure of the entire proteome, which is generated that you get. So that gives you an idea of the rate, and because the differences are so important, I think it's okay. But it's true that one would want to see more sophisticated experiments like we discussed with the polysomes, and maybe an idea of the number of. I mean, it would be really interesting to compare the number of ribosomes between the two species, of course. And there's still a lot to do in the experiment.
Yeah, I'm not sure if it would actually change the outcome. I guess I'm just kind of curious. It seems like the difference between like human and mice, it seems like different enough even on a mass specific basis that I'm not like it doesn't seem like it's kind of right on the edge. It seems like pretty clear, but yeah, I don't know. I'm just kind of curious like if there's a it seems like more of a decision, like a methods kind of decision of like, okay, is this method like sufficient, or does this actually like show what we want to show?
Yeah. Again, as I said, it's crude, but as you said, the result is quite. I mean, the difference is quite marked, so I think it's significant. So that doesn't mean that we should use more refined approaches to better characterize the difference.
I guess kind of related to that, there's almost like a meta question, but in like a lot of your guys' research, do you constantly face this kind of challenge of like the point of diminishing returns? Like it seems like there's always like some better method around the corner or some more like sophisticated design, and are you faced with the challenge of like, well, it's like this is really kind of one self-contained idea, and I should really kind of stop here and not worry about like chasing the next kind of iteration. Yeah, I'm curious how you guys navigate that in general.
That's more of a philosophical question, but it's an important one in science, I guess. Yeah. Now, I'm not always chasing for the the newest method, and I think there are some old and robust methods that work very well, and so here we used some of them. And there are of course some new methods that you have to use. I mean, I'm thinking about all the single cell omics, for instance, and which is really changing what you can do. But it's like computers. I mean, if you always wanted like the most advanced one, you never get the work done. Or so
students need to graduate before they retire too, so there's always that.
Yeah.
Well, this was an extremely exciting discussion. Thank you so much again, Brad, for leading, and Olivia for the wonderful contributions. It's always great to get the inside scoop on what went into a publication, because you really just get a snapshot of the final outcome. So it's a really unique opportunity. So we love hosting the journal clubs, is to meet awesome scientists, but also just kind of hear the perspectives that really sometimes don't make it into the paper for whatever reason. Kind of helps humanize the whole process as well. So thank you so much, and wonderful work. We appreciate especially your time today, and please reach out if we can ever be of any service. We sometimes have faculty posting lab openings in our Slack, or if you ever send us an email if you're hiring or recruiting students. We have plenty of Boston local folks, especially that are part of the bio community. So we'd be happy to share any exciting news with the rest of the group.
Okay, I appreciate it.
Yeah, of course. And
Thank you. Yeah,
Thank you.
nice to meet you all. And, yeah, see you around.
you.
Bye-bye.
Tend to live longer.
Kind of jump in and sort of lay out the groundwork for
setting up to do an experiment. You're thinking about all these different questions, and what made you decide on making this particular clock in this way?
So no, we did not design the clock. So we identified it quite a while ago, actually, in just by looking at chicken embryos, and it's now there are.
Like mice live like 2 to 3 years, humans live 100 years. I thought there'd actually be more substantial differences between the two, but it was kind of interesting that it wasn't as great of a difference, at least from my naive perspective, as like not in this field of study.
Another striking example that people use often is to say that the heart beats the same number of beats in mouse as in human, knowing that a mouse has a lifetime of 2 years, and human is about more 80 years. Means it beats much faster.
I think this is where kind of like anomalies or animals that don't kind of fit nicely along this model are interesting cases to pinpoint. Like that's are very to me are very interesting. And heartbeat is one, like they have a much faster heartbeat based on what would be expected for their body mass, and there's very clear like life. I guess
it's faster, so
there's no cell contact
that need to happen just happen more slowly on their own timelines, and aggregate of those things causes the overall development time to be slower. But
forming conversation, or if you just want to, we're going.
I read.
Else, like differentiated to this stage where you have oscillations, and you culture it in isolation. They will, if you treat them with, in fact, a YAP inhibitor, you will, so the cells will oscillate, and they will oscillate with their normal period, and you can do the same experiment also with mouse cells, and so which means that, and that's in complete isolation. And we don't understand how this can work because these oscillations are supposed to be driven by notch, but the experiments I'm telling you about, there's no cell contact, and yet they keep the endogenous period, and so that's something that is still quite mysterious. But that's why we are not so surprised to see that in chimeric conditions they would keep this period, because it seems to be really hardwired in the cells.
I have a technical question here. So in terms of the culturing conditions for the two different types of cells, were they optimized for like what would achieve like the best, I would say, like proliferation growth happenings of the cells for in the individual, the two cell types, or was it like how were those conditions chosen? Because that actually makes a huge effect of just like media and quantity of what you add to the media and proliferation rate, even just like in traditional cell lines. So I'm curious, like if for something that's like super time specific, how you sort of like make sure you had the right paradigm or the right composition for that.
Oh, that's a very good question. The whole difficulty actually of this was to find identical culture conditions that would work for both species, because if you start to play with the goodies and particularly the nutrients